get used to the interface. So I'm going to talk about a project that we have been working on for quite some time. It actually the project can be traced back to my PhD training. So I have a lot of deep interest in this problem and excited to share this story. So just for the little background, so get to especially the early year graduate student on the same page. So this as we know, a lot of cancer are caused by the fusion oncoprotein. Essentially, for example, in this very famous example, the so-called Philadelphia chromosome, essentially the two chromosomes apparently break off, and then they're the fragment of our chromosome, they're fused together, generating the aberrant and a cancer-specific fusion oncogene. For example, BCR-ABL, that's the cause of chronic myeloid leukemia. And we also know in acute myeloid leukemia, about 3 to 5% of this type of leukemia, including myeloid and T-cell acute lymphoblastic leukemia, they're actually caused by the fusion of NUP98, which I'm going to get into in a second. And as you can see here, the patient with this type of leukemia, they tend to show really poor prognosis and outcome in the clinic compared to the patient without this kind of a fusion oncogene. And transcriptome-wise, we also know why that is the case. When you're looking into the patient sample with the NUP98 translocation, you can see this leukemic patient sample express a lot of the so-called leukemic stem cell signature. For example, the Hox gene and MIS1 and PBX and those so-called proto-oncogene for leukemia, which I'm going to touch on several of this locus later on. And that's part of the reason why this leukemia patient with the NUP98 rearrangement show really bad prognosis. And that's the reason why we need to study this leukemia, so that we can understand what's the reason and how we can treat this patient. And I haven't tell you what is this gene is about. So for your background, normal NUP98, it's essentially a gene encoding a co-component of the so-called nuclear Paul complex, MPC. As you know, MPC, they're essentially situated on the nuclear membrane. That's the channel, basically trafficking things and out of the nucleus, including protein and mRNA, etc. And what happened to this leukemia patient is this N-terminal part of this gene of NUP98, they essentially break off abruptly and fuse to one of the many fusion partners. As you can see in this summary from a review I pull out of from the literature, 1/3 of this fusion partner for NUP98 are essentially transcription factor. They're bound to DNA, using this homeo box domain to bind a specific motif in the genome. And another third part of the fusion partner are essentially the chromatin binding factor or chromatin modified proteins. Some of them, for example, PH23 and KDM5A, they have the pH finger that can bind to the H3K4 methylation, especially tri and dimethylation. As you know, those marks are a prominent mark for the gene activation. You can see them at the promoter and enhancer regions. And the last part of third are mainly associated with the transcription, and some of them are actually understudied. And, as you know, the normal NUP98 show this classic nuclear envelope staining, but we know, the field know for a long, long time is this aberrant nucleotide fusion oncoprotein, they show, absolutely very different staining pattern. It doesn't matter what the fusion you're looking at, and it doesn't matter if you're looking at the primary leukemic sample, you can see, this puncta formation in the nucleus instead of this classic nuclear envelope staining for the wild type. So there's drastic differences between the non-nucleotide and the nucleotide fusion we see in the cancer. And we also know this puncti or condensate formed by nucleotide oncofusion proteins, they're Chromatin bound because of, as I mentioned earlier, a lot of this fusion pattern, they either bind to DNA or chromatin modification. So you can stain them, and you can see the fusion protein, the puncti, follow the histone, in this case H3K4 translation, very nicely. And we do not see this very distinct patterning of this histone modification in the normal cell, which I didn't show you. And suggesting actually the nucleotide fusion can reorganize the chromatin modification and potentially the 3D structure of the chromatin as well, which I'm going to talk about later a little bit more. So for a long time, we and there's a few that, are very interested in a set of questions I listed here. So first, what is really drives the formation of this puncti or false condensate I just showed you, which is essentially contained and formed by nucleotide oncofusion proteins. And this condensate are fairly big. They're ranging from 50 nanometer to 500 nanometer in diameter. So it's quite big. So what is makeup of this puncti in the nucleus? And then how exactly the condensate formation are related to the proto-oncogene deregulation we see in the patient sample, and how that is related to leukemic development. So this is a set of questions we started to ask, and then this project was initially spearheaded by a really talented postdoc fellow, John, who's now an independent PI in South Korea, and more recently he moved to industry. And then later on, this project was joined with excellent PhD student Iran, and he recently graduated. And along the way, we collaborated with a lot of excellent investigators, including Ben Sabari in UT Southwestern for the condensation study. For the 3D genome profiling, we have collaborated with Dr. Feinstein at the UNC Chapel Hill. So for the first question, what drives condensate formation? As you notice, the commonality of all these fusion oncoprotein contain nucleotide is really, the N-terminal region of, all these fusion proteins, as I'm showing here in this red boxes, right? And if you, run this sequence through, software, this essentially tells you this is a low complexity domain protein region, the so-called intrinsically disordered region. And if you dive into what is in this so-called IDR, it's really, about 40 of the motif, the so-called phenylalanine glycine repeat, the FG repeat sequences. And the idea has gained a lot of attention in recent years. You probably heard a lot of them, and some of a lot of this idea actually have the patterning in terms of the amino acid composition. I just list here some example on the left, right side. For example, the polyQ. You heard from, probably this is a motif associated with neurodegeneration and other diseases, oppositely charged blocks. And for this particular idea within the nucleotide, it's different. It essentially has, a number of the hydrophobic and also aromatic residue kind of sort of spreading out across this region, about 500 amino acids. And we call them a uniform distributed aromatic residue, or UDA. I'm going to come back to this later on in my summary slides. So what John did initially, he just purified the recombinant protein from bacteria using this common region of NUP98 that is retained in all the fusion that we see in leukemia patients. And then, as you can see, when he used this recombinant protein, he, played this purified protein on the slides. You can see the beautiful condensation formation. And this foci, you can see the formation of this foci really depend on not only the concentration of the purified protein, but also the number of this FG repeats that he included in this recombinant protein. As you can see, when he reduced the FG repeats number from somewhat 40 to 20 to the tens, and he sees dramatic decreased formation of condensation. And we can also use a different strategy in which we mutate FG repeats to the syringes or AGs. In a way, we change the aromatic residue to the non-aromatic residue, right? And we can recapitulate the requirement of FG repeats for the condensation formation, both in vitro, which I didn't show here, and also in the cells, which I did show here. As you can see in the cells, when we introduce the fusion with the intact the FG repeats, you see this puncta formation. But when we mutate to FG to the syringes, you see this sort of a smear, but still in the nucleus. And we can use different mutation, for example, alanine G repeats. And this kind of a requirement for aromatic residue and potentially multivalent interaction is really consistent with a concept, the so-called condensation or liquid-to-liquid fast separation. And there's a lot of literature and review article, really wonderful review article that cover this space really, really well. And I suggest the student in the audience can refer to this review and an opinion article that are listed here. But just for simplicity, I just want to say the FG repeats can mediate not only a hydrophobic interaction, but a bipartite interaction. You can imagine, you have a two protein, and each protein have 40 different ligands to form, interaction between themselves, right? So if, this, hydrophobic interaction or bipartite interaction deassociate, there can quickly, this ligand can quickly reestablish the interaction with any of the other, 40 potential interaction partner, between themselves to regain the association. So in a way, the rate of deassociation, is really less than association. So in a way, this multivalent interaction form the condensation, and that's the basis for this phenomena. And that's the reason when we disrupt the FGE repeats into the syringes, we essentially, lose the condensation formation by these proteins. So then, let's move on to the next question. What is the makeup of this, condensation made by NUP98 fusion protein in the nucleus? To that, we use a proteomics approach, and this bio-ID or turbo-ID probably used and or hear this technology. Instead, in essence, what you do is you fuse the protein of you're interested in this case, the NUP98 fusion oncoprotein, we're studying, to this Bure ligase, which is to induce biotinylation to the potential interactors nearby. And then you can, purify the biotinylated protein for mass spec analysis. And because we know the FG repeats or the UDA, uniformly distributed aromatic residue is critical for, condensation formation. And so we essentially included a mutant as a control where the FG repeats now mutated to the alanine genes. And so we did this proteomic study, and then the result really both surprising and exciting. So essentially, what you're looking here is a plot. You compare the intractome identified using the fusion protein containing the wild type FG repeats compared to the mutant where the FG repeats are now being mutated to the AGs. And you see the enrichment in the wild type fusion, which means you have FG repeats, but not compared to the AGs. And you see, the several classes of complex has been pulled out, which is enriched in the wild type unconfusion, which includes, notably the so-called ML complex. You can see ML1, ML2, WBR5, and RBBP5, and they're, highly enriched. And also you see the so-called Swan-Sniff complex. These are, remodeler that can open the chromatin to promote the DNA accessibility. And they're considered to be the transcriptional coactivator. And we also see the histone acylation related factors. For example, the P300 and this is a TIP60, P400, one of the largest subunit of the TIP60 complex. And also BRD4, which is, as you know, a acetylation reader protein and a quite important coactivator as well. And because, we see this interaction, we use a lot of different way to verify this interaction. Long story short, we can verify the indeed form interaction with wild type FG, but not the mutant FG, which we use AGs. And I just want to give you one of the experiment and the related data. Here we use a so-called Le Carré system, a reporter system. And in this system, the cell has been engineered with the integration of a rather large lacquer repeat in one particular chromosome regions. And the way this works is when you fuse the protein of your interest in this case the ID of nuclein 88 to like I, this protein bind to this repeated sequence, we can imaging this fusion protein because the fusion protein is also fluorescent labeled. And on top of that, if you do the staining for the potential co-activator or partner that has been recruited or into this foci, then you can see, the change of the color. So that's, the quantification method for protein-protein interaction. And that's exactly what we did. For 3 coactivator, we identified from the BioID analysis, as I mentioned early on, which includes the ML1, one of the methyltransferase of the ML-WDR5 complex, BRD9, which is a Swan-Sniff component, and a WDR4. And in all cases, we can see the FG repeats when fused to this LAC system. We can see the recruitment in all these 3 different cases. And then when we mutate the FG repeats to alanine genes, we essentially set this interaction or recruitment back to the basal level. You can see compared to essentially the just empty vector, just the DNA binding domain itself. And in this, the nice part of this system is we know exactly where this locus of lac repeat is located. The locus is actually located in one of the subtelomere region, known to be very much heterochromatin. Because if you stand for H3K9 translation, you can see, it's really lighted up, but which is in contrast with the lack of the active chromatin H3K4 translation and histone acylation, right? And then we ask, what happened if we, recruit the FG repeats from nucleotide into this locus, and then, you can see we see very dramatic chromatin remodeling effect. In essence, the FG repeats, when being recruited to this locus, it causes the dramatic reduction of H3K9 translation, which existing in this locus naturally, right? In the backgrounds in the background cells, and simultaneously we also see the gain of H3K4 translation and histone acylation. And because this is just one reporter locus, we also use the cell which we ectopically express the unconfusion either with wild type FG repeats or mutated FG repeats, and then perform the genomic analysis. And you're looking at the Hox locus and A or Hox B locus, which is a classic oncoprotein of this disease. And you can see when you mutate the FG to the adenine genes, the binding of the ML complex is reduced, and so is H3K4 and histone acylation decrease, as you can see. And not only just these two locus loci, if we combine other peaks, the region that are bound by unconfusion, we can also see the similar effect. That means the recruitment of ML complex and increase of histone activation related marks are both depend on the FG repeats of this fusion. And so, just to put everything together, so using the in vitro system, the very first part of my talk using the recombinant protein, we can establish the puncta formation. And because in that system, we just use the purified protein. So the purified protein of NUP98, essentially this region enriched for FG repeats, is sufficient to form the condensation. So we call the homotypic interaction. This homotypic interaction is essentially drives the condensation, forms the puncti. And it is, another type of interaction, the heterotypic interaction, essentially FG repeats and a coactivator interaction. And this interaction can induce the recruitment or co-partitioning of the coactivator into the puncti. And because the fusion partner of NUP98 a lot of them are DNA binding protein or histone modification binding proteins. So essentially, that's the reason we see this puncti associated with chromatin. And as you know, the collective action of all these factors, including the ML and histone acetotransfers and potentially, the Swan-Stiff complex can drive the high expression of the target protein, target genes, and a lot of them I just mentioned, for example, the Hox genes. And this is, one of a more detailed diagram of what I just said. And in an early study, we also show the IDR can induce the chromatin looping. And this, the story was published a few years back. And, for the sake of time, I just want to mention, you can see the unconfusion bind to, different regions, either promoter or enhanced region of this PPX3 genes. And using the high-C mapping technology, we can see among any two of this binding site, we can see a looping formation. And because this locus does not have CTCFs, this is really the CTCF independent chromatin loops that are formed by the fusion. And in the last slides, data slides, I just want to mention, the FG repeats are absolutely critical for oncogenesis, both in vitro, as you can see, when using different oncofusion, when we mutate FG repeats, we essentially lose the transformation, and we also can recapitulate the same phenomenon in vivo as well. So, come back to, I have 2 minutes, take home again, what happened in this fusion oncoprotein, the genetic mutation essentially, generated a fusion which contain the low complexity, region. And in this case, the FG repeats, as I mentioned, UDA, right? This cartoon stands for, again, the aromatic residue that has been distributed in this region. And this motif UDA has a lot of power, had a lot of force. One side, using the self-self interaction, it can induce condensation, and simultaneously through the self and the partner association, it also can recruit and partition the coactivator. And on top of that, it can also rearrange the 3D Chromatin genome. I know I, speak very fast, and I list some of the paper we published. So if you're interested, you can dive in. You can refer to this papers. And in closing, I just want to also sort of speculate. The UDA, this motif, is probably going to be very important, not only in the malignant settings, but also going to be important for normal settings. For example, as we know, the RNA polymerase 2, you have this typical, motif, haptopeptide sequence. And the first two residues, Y is essentially very much like FG. So I propose, this UDA sequence or code going to be fairly critical for transcription activation, and there's a lot of more work to be done. So I run out of my time. I'm just gonna closing